This is a video about the Central Dogma, in other words, DNA and protein synthesis. Here to help you learn about DNA, we have Eloise, Kat, Liam, and Sasha.
The central dogma is the process in which DNA is converted into RNA, more specifically messenger RNA, and then furthermore how the RNA is used to form proteins. This process begins in the nucleus and ends in the cytoplasm of the cell. It is important to understand how your body creates proteins because they are the building blocks of one cell.
First, we will talk about the structure of DNA. We are using a ladder model of DNA, even though DNA is in the double helix form and is twisted. Double helix is a spiral consisting of two strands on the surface of a cylinder that coil around its axis. DNA is made up of nucleotides. Nucleotides are bonded phosphates, deoxyribose sugar, and a nitrogenous base. Phosphates are made up of and a form of phosphoric acid. There are four nitrogenous bases in DNA: adenine, thymine, cytosine, and guanine. The sides or backbone of the ladder are the nucleotides, and the rungs of the ladder are the nitrogenous bases bonded by a hydrogen bond, which is an attraction between the two positive and negative poles of charged atoms. The two strands of DNA are anti-parallel, meaning that the head of one strand is always laid against the tail of the other strand of DNA.
Now we will talk about transcription because this is the first step in the central dogma. Transcription is the act of turning DNA into RNA. Transcription occurs in the nucleus of a cell and starts out when the promoter, a region of DNA that leads to initiation of transcription, is bounded by RNA polymerase, which is a catalyst that separates the DNA. The double-stranded DNA now becomes single-stranded. Then one of the strands of DNA uses the template from which RNA nucleotides assemble into complementary strands of RNA. However, a compound uracil takes the place of thymine in RNA. Then the mRNA, which is the RNA that carries genetic instructions from the nucleus to the ribosome, is edited. This happens when bits and pieces are cut out. The portions that are cut out and discarded are called introns. The remaining portions, known as exons, are then spliced back together to form the newly edited mRNA. Now the mRNA is ready to travel outside the nucleus to the cytoplasm.
Then the process of translation begins. The mRNA strand exits the nucleus and travels into a ribosome, which is an organelle consisting of RNA and associated proteins. The tRNA, which is RNA that carries amino acids from the cytoplasm into the ribosome, that has the anticodon, which is a sequence of 3 nucleotides forming a unit of genetic code in a transfer RNA molecule. Each tRNA corresponds with a codon, which is a sequence of 3 nucleotides which together form a unit of genetic code in a DNA or an RNA molecule. Each transferred DNA links with its corresponding mRNA. Then, tRNA connects its amino acid to the polypeptide chain of other amino acids. The strand of mRNA starts to feed through the ribosome when it reaches the start codon, which is the first codon of a messenger RNA transcript translated by a ribosome. The start codon always codes for methionine in eukaryotes until it reaches a stop codon, or termination sequence, which is a codon that signals a termination of translation into proteins. Then, the tRNA zooms back off into the cytoplasm to pick up another amino acid and repeat the process again.
Polypeptide chains are the result of translation. Each polypeptide chain is made up of a series of amino acids joined together in a specific sequence whose function is dependent on its chemical structure. The primary structure of a protein refers to the sequence of amino acids in the polypeptide chain. The secondary protein structure is hydrogen bonding of the peptide backbone, causing the amino acids to fold into a repeating pattern. In this structure, there are 2 types of folds, one being the plated sheet and the other being the alpha helix. The tertiary structure of a protein is the 3-dimensional folding pattern of a protein due to the side chain interaction. The quaternary structure of a protein is the protein consisting of more than one amino acid chain. If the polypeptide chain does not fold and coil into the correct protein shape, the chain will die and the process will restart.
This process is important because without it, we wouldn't be alive. The process of central dogma creates protein. Protein has many crucial functions, such as creating hormones and helping our body defend infections and illnesses. If there was no central dogma, we wouldn't be able to function. The central dogma is the key to life and existence.